Hi, simply in this video I am going to talk about how the drugs enter the cells. This is a part of pharmacokinetics lectures and I'd like to use which color? Okay, blue. This is the cell and this is the medicine. Now how he going to enter this cell? When entering this cell, the main obstacle he have to pass is this cellular membrane. We know the cellular membrane has lipid bilayer and proteins embedded in it. The first pathway we are talking about is passive diffusion through the lipid bilayer. And then we are going to focus on carrier mediated transport which occur through protein carriers. Then aqueous pores and endocytosis, which is take these drug substances and make a vesicle and ingest it into the cell. From these aqueous pores and endocytosis, I keep it aside because it is not much important, and focus on passive diffusion and carrier mediated transport. Passive diffusion, let's erase, passive diffusion. In passive diffusion, the drug crosses the cell membrane by the lipid bilayer. For this, there are three factors that are affecting the diffusion. This always occur along a concentration gradient. The factors that affect this passive diffusion are the lipid solubility, degree of ionization, and molecular size. The lipid solubility, it decides the absorption rate and whether it is crossing the blood-brain barrier, because lots of lipid soluble medicines dissolve in the blood-brain barrier but others do not. Therefore, if we need our drug to be bind to a target in the CNS, brain and the spinal cord, we have to cross the blood-brain barrier. Therefore, we should select a lipid soluble drug molecule. Then let's move to degree of ionization. For this, I have given an example which make it easy to understand. I have take a weak acid and a weak base because most of drugs are weak acids and weak bases. Moving on to weak acids, I have taken HA is dissociating into H plus and A minus, which are the ions. The HA is non-ionized form and A minus is the ionized form. H plus is for the pH. The common thing we have to remind in this place is that this cell membrane permits only the unionized form to go through the lipid bilayer. The ionized form is for carrier mediated transportation. Unionized form is for lipid bilayer passive diffusion. So weak acids, when they are put into a acidic medium according to Henderson and Hasselbalch's equation, when pH reduces, when it becoming an acidic medium, the weak acid in the acidic medium do not dissociate it, because this is H plus and we are giving more H plus in the acidic medium, so the equilibrium goes to this side, left, and forms more HA, which is the unionized form. Therefore, unionized form is increased, which can cross the lipid bilayer very easily. So they are absorbed. They are well absorbed. So the reverse is under the weak base. In weak base, this is the ionized form and this is the non-ionized form. So as we have on the previous example, we have take the weak acid into the acidic medium for good absorption. Therefore, for the good absorption in weak base, we have to put this into a basic medium. Therefore, this when the basic medium has OH minus, this equilibrium is biased to this side, right. Therefore, B is increased, which is the non-ionized form of weak base. And non-ionized form always crosses the lipid bilayer easily, while the ionized form do not. Then the molecular size, it is not much important. I have missed. Yeah. On the degree of ionization, we come across these two terms, pH partition, and this is ion trapping. pH partition and ion trapping. Firstly, let's focus about pH partition. It is like this. We know that weak acids are absorbed in acidic medium, and the weak bases are absorbed in basic medium. If weak acids are absorbed in acidic medium, what happens if it is in basic medium? We are just investigating. In that place, it do not forms the unionized form. Therefore, it can't cross the cell membrane because it is ionized. Therefore, it accumulates outside the cell. For example, when excreting a drug through the kidney, in the kidney tubule, we have the weak base along with the acidic urine, then it will be ionized, and ionized form do not transported through the cell membrane. Therefore, this cannot be taken into the renal tubular cells. Therefore, if it is not reabsorbed in the renal tubules, it is happily excreted from the body with urine. So, it is a good aspect that weak base drugs cause acidic urine. What is ion trapping? Ion trapping is we are giving the unionized form of the drug, and that unionized form, when crossing through the cell membrane, it is ionized. It is converted into ionized form. What happens? On ionized form, it was easily go through the cell membrane, but ionized form do not go through the cell membranes. Therefore, afterwards, the transportation of this drug is question mark. For example, aspirin. Aspirin is a good example for this, because aspirin once taken into the cell, it do not go into the bloodstream very quickly because it is now ionized. Therefore, it stays in the stomach and then inhibit the cyclooxygenase and decrease the production of prostaglandins, and the inhibition of gastric acid secretion by prostaglandins collapsed. Therefore, gastric acid secretion increases, causing gastritis and all those things. The acidity of the stomach increases. So, that is a complication. Okay. Now, what's left is this carrier-mediated transport. It is two types, active and passive. Active is using energy, passive is not using energy. Active is this carrier-mediated form always occur through the proteins and mostly probably water soluble things, and ions go through these protein pores. Active and passive here. And active process happens against the concentration gradient, while the passive form acts along a concentration gradient. These things are very important in blood-brain barrier, biliary tract, and renal tubules, including the intestines. So this is a nice short note about the transport through the membranes, pharmacokinetics. I hope you have get something from this video. Thank you.